Hello grade 12s and welcome to Life Science, and today we're going to have a look at genetic engineering. Okay, so let's go straight into it. Okay, first of all, terminology. Okay, what do the following words mean? We need to have a look at it. Okay, so first one, of course, is mutation. Then I want you to look at mitochondrial DNA. Okay, and then I want you to give me genetic engineering. Okay, now there's three words and there's quite a lot that you can tell me about them. So I'm going to give you 3 minutes to quickly find out as much information as possible on those three words, and you're going to give me everything when we get back. Are you ready? Off you go. Welcome. Let's have a look at it again. Right, I'm sure you've given me everything that you have. Let's see. Here, mutation. It's a change of the structure of a gene resulting in a change from which way the terminated, well, let me get this right, in which may be terminated to the next generation or passed on, transmitted. See, somehow I can't do it. It's transmitted to the next generation caused by alteration of a single base unit of a DNA, or the deletion, so in other words taking out, or the insertion or rearrangement of a large section of the gene or chromosome. Now, you got to understand, now remember when we have to look, or when we do protein synthesis, you're going to learn about your DNA structure and things called like thymine and adenine and guanine and cytosine, and they've got to be in certain orders to create certain things, right? Now, if one of those aren't in a certain order or something is missing in that gene, you're going to change, okay? So this is where it comes from. It gets passed on from generation to generation, okay? And if something changes, so let's say one thing is left out, that will mutate. It will make that thing mutate into another gene, okay? Then we get mitochondrial DNA. This is quite a cool one, okay? It's DNA found only in the mitochondria and is inherited from the mother. Now, here's a small question from you. It was a question that they asked once upon a time in a matric paper, okay? They said, mitochondrial DNA, why is the mitochondrial DNA only from the mother? Why can't the father give it? I mean, think about it. The dad is part of it. The dad gives his sperm, right? Which means he should give DNA, okay? Now, think about this very carefully. When the egg and the sperm come together, okay, the mother has DNA in the sperm, okay, in the egg, right? And it's got mitochondria to give it energy. The sperm has got its DNA, right? And it's got mitochondria on the inside as well. And it swims because mitochondria gives it energy. So it swims all the way to the egg. And when it gets to the egg, it only allows the DNA to go through. The rest of the sperm stays outside of the egg. So does the male's mitochondria ever enter the female? No. So our DNA of a male goes into the female's egg, and the female's mitochondria stay behind. So in actual fact, you can trace back your ancestors through your mitochondrial DNA. But you can't just do your ancestors, you can do your mothers, the female part of your ancestry. Hey, how cool is that? That's quite cool. Hey? Next one, let's have a look. Genetic engineering. Okay, now genetic engineering is the very, very specific thing. Okay, it is the modification of characteristics of an organism by manipulating its genetic material. Okay, now we change the genetic material. That's what genetic engineering actually is. Okay, so we change the genetic material so it suits what we want. Okay, for example, if we want to change things, I'm trying to make it as nice as possible, because I don't want to bring the main one in yet, okay, that we look at. But let's say we want something sweeter. So we have a look at certain ways to change its genes to make it sweeter, or we want it bigger, or we want to change it to make it, let's say the millies of the land to be more drought resistant. So we're going to change it. We're going to genetically change it to make sure that it does exactly what we want. Okay, one of the nice things we do is we change roses. How cool is that? We genetically manufacture or genetically engineer roses. Because have you ever gone to a nursery or where you buy roses? Have you ever bought rose seeds? You can't. They come from cuttings. Okay, so what we do is we take a cutting and we genetically modify them. We genetically modify their cells to create what we want. Okay? How cool is that? That's genetic engineering. There's the words that I've asked for. Genetic engineering, mitochondrial DNA, and mutation. Cool, eh? Now, genetic engineering, if we go straight into that, into your genetic engineering, okay, there's a couple of things that we mess around with, okay? The first that we're going to have a look at is this thing called stem cell research. Now, before we go on to what stem cell research is, what is a stem cell? Okay, a stem cell is a cell that can become any cell in your body. Okay, it becomes any cell in your body. Remember, we spoke about how cells, there's a certain cell and they get old and they start going through differentiation, right? Now, these cells can become anything. So, for example, if I want heart tissue, okay, and the heart itself, or let's just say the heart reproduces heart tissue all the time. The muscle in your bicep does muscle all the time. Your esophagus, when it needs to be fixed, it creates esophagus cells. Can you understand? It makes a specific cell, so it's part of that tissue, right? Now, a stem cell can be any single cell. How cool is that? Now, that is what is so important about stem cells. Now, stem cells, you normally get it from your umbilical cord, for instance. Okay, so one of the big projects is at the moment is that when you have a baby, the umbilical cord gets kept and you send it away, and that's where you find all your stem cells. So one day when you're very sick, you can, and you need a liver or a kidney or whatever, they can grow it from the stem cells. How cool is that? Then you don't have to be on a donor list. Hey, that's quite a cool one. Now let's have a look at what stem cell research is about. Okay, now stem cell research on the stem cells and the use in medicine. That's exactly what stem cell research is how we can use them to make things that we need all the time, like liver, like kidneys, like hearts, okay? Because these are the ones that people are they're waiting on a transplant list and they can't get to it, okay? So the quicker we do that, the better, okay? Next thing, genetically modified organisms. That means that we genetically modify them to what we want. Okay, so let's see. An organism whose genome has been altered by techniques of genetic engineering so that the DNA contains one or more genes not normally found there. So for example, one of our genetically modified organisms, this is a cool one, is insulin. Insulin, we modify insulin. Okay, now we take a specific bacteria, and that bacteria is E. coli. I just had to think of the name now. Now, E. coli, what E. coli does is very bad for us, but E. coli also makes insulin. So what we do, let me just check if I've got it here. So we're actually going to talk about two things at once. Let me move on to the next one rather before I explain exactly what we do with the E. coli, okay? So we genetically modified organism. We modify an organism to suit us, to make it easier, okay? The next one is biotechnology, okay? This is the use of biological processes in industry and other processes, especially with genetics, okay? We're multiplying or we manufacturing and we making microorganisms for the production of antibodies and hormones, okay? So antibodies and hormones, that's why I stopped, okay? Antibodies and hormones, there we go, that big word there. Now, what we do, if we take this bacteria, right, and we open up its nucleus and its DNA and we have a look at the part that makes the insulin, right? And we take that insulin out. We take that insulin out and we stick insulin in there, it makes new insulin for humans. But if we take a human insulin gene and stick it in there, it's going to make human insulin. Now, if you think about it, in the old days, okay, very long ago, they used to use pig insulin because a pig is very similar to humans, right? A lot of their organs inside are the same. Okay, so we used to use them. The problem is, okay, there's a lot of things that's bad about using a pig. First of all, the biggest thing is that there was huge side effects. People got sick. It didn't last long. It didn't help much. Even though they were getting the insulin, it wasn't good insulin. It wasn't good for us, okay? Another one is religion. Okay, so a lot of people can't have pork in your religion. Okay, so if you're not allowed pork, how are you ever going to get insulin? How are you ever going to get that to protect yourself and keep you alive? If you can't go near pork, it's not going to help you, is it? And that's a little bit unfair. Okay, so what they've done is they needed something that we could play with, right, and make insulin. What is the quickest thing that reproduces? One of the quickest things is bacteria. So let's take a bacteria, we change its DNA structure, and we make it reproduce. If it reproduces, it makes lots of itself. If it makes lots of itself, it'll make more. And the more it makes, the more insulin it makes. That's how we get our insulin that we can inject ourselves with. And insulin is a hormone. Okay, so that's how we use it in biotechnology. We take genetically modified organisms and we use it in biotechnology. Can you see how you got to put two and two together in this? Okay, very, very important. And then we have another thing over here that we do use. It's called cloning. Now, we can clone. We've cloned Dolly the sheep, for instance. Okay, now remember that your body clones all the time. Okay, your body clones every time you cut yourself. Your skin has to make new cells, so it's cloning itself. It's making a new one of you. Okay, so if a cell clones, it makes a new skin cell. Okay, now cloning is a cell group. It's a group of cells or organisms that produce asexually from a genetically identical to a single ancestor. So in other words, if I say to you, an amoeba, a single-celled organism, it goes through it goes through mitosis, splits, and now there's two amoebas. Okay, that is cloning. Now this one is going to split again. This one's going to split again. It's cloning. It's making itself again. There's no genetically, there's nothing different, nothing genetically different between the two. Okay, and that means that if I had to clone myself, which is illegal. Okay, so if I had to clone myself, I will look identical. Okay, I would look like I have a twin. The problem is, it's not going to mean that if I clone myself, is going to be as big as me immediately. It's going to start off as a baby. Okay, and it's going to grow up, and it's going to look at my age that I am now. It's going to look the same as what I am, except if I've lost a finger. Okay, and he hasn't. Would he only have 9 fingers if he comes out, or will he have 10? He'll have 10, because genetically I have 10. If I lose a finger, I didn't lose my genes for the finger. Does that make sense? Right, so now that I've given you all this information, right, and I can see the smoke coming out of your ears, and it's all going in and it's settling, I think you need a break, because I sure do. I'll see you now. Cheers. Welcome back, and now that I've given you all this information, I've had a look, and I've came to the conclusion that there's a lot of things that we haven't looked at, and we must look at problems when it comes to our genetics, right? And one of the biggest things that they like looking at, okay, is blood. Okay, so what I've done here is I've given you one of these blood groups things, okay? Now, you're going to do it, okay? But I want to show you a couple of things first before we do it. Have a look at the board here. Don't look at the sum. I'm not interested in the sum. And you know I've put that out because that's how I want you to set it out, so don't forget. Okay, what I'm doing is I just want to add a little bit more space, if it'll allow me today. There we go, a little bit more space, okay, so that we can talk. Now, come on, just think about it. What type of blood group do I get? Okay, I know that you know that we get A blood group, okay, and we get B blood group, and of course we get O blood group, and we get AB blood group. Okay, so those are the blood groups we get. Okay, now when we have a look at this, you need to understand this is phenomenal, because we have certain things that are so important about this. We have a thing called codominant. We have a thing called complete dominance, okay, when it comes to blood. Okay, so watch this. Remember, I always tell you that you need 2. One from your mom, one from your dad. So if I say you are A, that is your phenotype. So let me just erase that again, okay, and I'm going to put here, that is your phenotype. Your genotype, on the other hand, is 2 things. So your genotype for this would be A, right, or something else. So it's that or something else. B would be B, or something else. O would be O, or could be something else. And AB, or could be something else. Now I've put the ors for all of them so you can see what I'm saying. When it comes to blood, we know that A is dominant over O. So if I have this, if I have AO, what blood group is it? It's A. I look at the dominant. Okay? If I have B, I can have BO. So I know which is dominant, which is the O. Okay? Now, can I have anything else but OO? Can't. Remember, to be able to see the recessive gene, you need both the recessives there, right? And of course, lastly, with AB, it can only be AB, it can't be anything else. So when it comes to blood, this is very important. Now, I like doing this, okay? But there are different ways of writing it. You'll notice that we write it like this, where you got A here and A here, okay? You've also got or, you have your 1 and you have your small one. Can you see how I've done that? Or here, it's B, B, or B, O. Of course, this is O, O. See how I've done it? And then, of course, A, B. That's how you, those are the two ways of writing it, okay? Now you've got both. They love this way. So please, I like this way, and this way is right, and this way is right. But they love both. Make sure you know them, okay? Now, let's get back to this. Here we go. I've got a genetic cross for you, okay? And I'm only going to give you 2 minutes, so make sure that you concentrate on this, hey? Okay, here we go. Could a man with B blood and a woman with AB blood produce a child with O blood? Okay, show all working out, and you have 2 minutes. Are you ready? Off you go. These were nice and easy. Let's have a look at it. First of all, what have they given you in the sum? Have they given the genotype or the phenotype? They've given the phenotype, right? The physical features. So let's have a look. The P1 generation, let's have a look at the genotype. Okay, it's either, if I have a look at the man is either BB, big B, or big B or an O. Am I right? Okay, of course, the woman is only AB. That is it. Can't be anything else. Okay? If I look at the gametes, and I've done it like this for a reason. If I look at the gametes, it's B and B or B and O. And then of this one, of course, it's A and B. Those are simple. If I bring it down, oh, look how many slides I've got here. Way too many. Way too many. So let's have a look. I've done it like this. I have either AB, there it is, AB for mom, okay? And then I've got B, B, or I've got B, O. So let's have a look. It's AB, AB. Oh, that A is terrible. AB, or it is BB. It is AB or BB. It is AB or BB, it is AO or AO. So, what can I deduce? The ratio genetically. Now remember, this is just to show you how things work. So I need to look at this one separately to this one. I just put two crosses in there so we can see what's happening. Okay, so the first ratio is 50% AB and 50% BB. Okay, and on the second one, you have, if you have a look at it, you have 25% AB, you have 25% BB, and you have 50% AO. How did I work that out? 25, 25, and together they make 50. Can you see that? Nice and simple, hey? And now, the last thing is phenotype ratio. We have on the top one, I have 50% AB and 50% B, because it's only the phenotype. And the bottom one is, let's see, 25%, 25% AB, 25% B, and 50% A. So, is there any chance? Let's see the question. Because at the moment, everything we've done up to there, everything up to there, has it answered the question? The question is, could a man with type B blood and a woman with type AB produce a child with a type O blood group? And the answer is no. You need to say, no, they cannot, because all they can produce, if the dad is BO, then this would happen. If the dad was BB, this would happen. Okay, so there is no ways that they can get that right. Cool. Okay, now let's have a look at the next one. I've got another one for you. Okay, here we go. Suppose a father and a mother claim they have been given the wrong baby at a hospital. It happens, trust me. Okay, both parents are blood type A. The baby they have has been given is blood type O. Can the baby belong to the parents? Can the baby belong to the parents? You have got 2 minutes. Are you ready? Off you go. Okay, let's have a look how you did. Okay, so the parent, let's say a father, they said they were both parents are blood group A. So let's do it this way. So we have A and A, or A and O, remember? Okay, and the same with the mom, A and A, or A and O. Okay, so we automatically know that we need two O's to create an O child. Am I right? Okay, so I'm going to cancel these ones, and I'm going to say the gametes that I'm looking for is A and O. Or should I say, should be a dot, not an O like that. I'm going mad today. Look at this there. Take that one out there. Let me put them back quickly. That's an A there, and that is an A, and that's a dot. Okay, dot, and the same here. It is an A with a dot, makes it an AO, and you're going to be ending up with, and you can see I've done both here. It's going to be A and a dot, and it's going to be A and a dot. Now you'll end up with an A and an A, same as what we did in the first place. Okay, and we're going to end up here with AA and AO. You're going to end up with A and O, and you're going to end up with two O's. So what is the ratio? If we have a look at it, we always put our ratios in. We have, okay, 75%. Oh no, this is the genotype. So we have a, let's see, we have a 25% AA. We have a 50% AO, and we have a 25% OO, which means the phenotype ratio is 75% A, or shall I put it this way rather? No, I can say A, that's right. 75% A, or, and we have 25% O. You get that? That's quite cool, huh? And what is the answer? Have we answered anything yet? No, we haven't. The baby that they have given the blood type O, can the baby belong to these parents? Yes, it most definitely can belong to these parents. Okay, I know you got that. You see how I use both ones? Let's see if you can get it, okay? Now, you need a break, I need a break. I'll catch you straight afterwards. Welcome back. Let's have a look. Now, we did the one with the blood. Another thing that they love looking at, okay, is sex-linked characteristics. Sex-linked characteristics. Now, if I have a look at sex-linked characteristics, I'm just going to move it down. Remember, I show you everything first. Come on, page. Let's extend. One more. You can do it. There we go. Okay. If I look at sex-linked characteristics, okay, so I'm going to talk to you before we get this thing. We all know we have these guys, oh, come on, these guys, and we have these guys, okay? Now, you know and I know that the XX is, of course, your females, and the XY is your males, okay? So we all know that. Now, please understand this, okay? Your X chromosome, yeah, a lot of the times, if an X chromosome has something on it, so in other words, some sort of disorder on it, when it comes to the male, okay, the Y can do nothing about it. Okay, so let's put it this way. If I say to you, this X has, let's say that 1 is dominant over 0, and hear me out. Let's say black is dominant over white, okay? And I say this X is black, and this X is white, okay? What is it going to be? This whole thing is going to be black. Remember, same as the color. Remember the big B and the small b? Can you remember that? It's exactly what I'm showing there. Now, look here. If I say the same thing over here as I was saying over here, let's just move this out, and I do the same over here. This has got black. The Y is not part of the X. What color is that going to be? It's going to be black, because the Y cannot mask anything. Or should I say, let's pretend that this is white. Okay? That means that this Y chromosome will not do anything to that, so it's still white. Okay, so the Y chromosome cannot mask the X chromosome. An X chromosome can mask an X chromosome. A Y chromosome could mask a Y chromosome. But can an X chromosome mark an X chromosome? Okay, it can't. If an X chromosome, can a Y chromosome mask an X chromosome? It can't. The X chromosome will always mask. Do you understand where I'm coming from? It's always a stronger one. And the best way, the way that I used to remember it, okay, and the best way I can tell you, and the best way I can think of making you remember it, okay, is if you have a good look at it, okay, the male is actually an X, and when it comes to the Y, okay, it is an X, it's just its leg is a little bit shorter, okay, so its X, its leg is a little bit shorter, which makes it look like a Y chromosome. Can you see the X there? Let me just change color here. Can you see over here? Can you see that little leg is still there? So it is an X, it's just a little bit weaker. Okay, that doesn't mean that men are weaker. That does not mean. Let's just put that out there from the beginning. That leg is just a little bit shorter. Okay, now, I want you to remember that very carefully. Okay, now, let's go on to the sum. Think about this carefully. Okay, a mother and father with normal color vision produces 3 male children. 2 whom exhibit red-green color blindness, and 2 female exhibit normal color vision. Demonstrate the inheritance of green-red color blindness in the male children. So, what they want you to tell me at the end of this is, how come the males are colorblind and the females are not? Okay, and it's very, very simple. You need to see this carefully. Okay, look here, a mother and father with normal vision. There's the key thing over there, normal color vision. Okay, I'm going to give you 2 minutes. Concentrate. Let's see what you can do. Ready? Off you go. Okay, let's see if you got it right. I'm going to go a little bit quick on this one. Let's have a look. Okay, so let's have a look at the mom, XX, and dad, XY. Okay, there we go. X1, XX. Okay, if a father has normal vision, okay, because it's normally a X-linked chromosome, there's going to be nothing on the dad. And let's say the mother has a C for color blindness. Okay, then let's have a look how this will work. Gametes will be a X with a C and an X. And the male will just have an X and a Y. Okay, simple. Bring it down. Here we go. X and with a C and X. And this will be X and Y. Okay, let's put this together. X, X, and that one's got a C on it. And this will be X, X, and this will be X with a C on it, right? And a Y, and X with a C on it, X with a Y. Okay, now, have a look at this carefully. Percentage ratio, we all know it's going to be 50% woman, 50% man, okay? And your ratio is going to be 25% XCX, okay? 25% XX, 25% X with a CY, and 25% XY, okay? And that means you're going to have 100% normal female, and you can have 50% normal male. Because look, soon as that one happens, becomes colorblind. So can you see how it works? It was quite cool, hey? Now, what I do want to go with you, want to have a look through, is this thing. A pedigree diagram, or a phylogenetic tree. Okay, now, I'm going to go through it with you. See if you can get this one, and hopefully I have enough time to give you one to do by yourself. Okay, so let's have a look at this. Here we go. Study the pedigree diagram of a family who have some individuals' hemophilia. Okay, the hemophilia is a sex-linked disorder. Use H for a normal blood clotting and small h for the hemophilia trait. Okay, there we go. There it is. Okay, so we know that female with white is normal. A female without, or let me just bring it down. Yeah, a normal female is a circle, okay? A male will be a square, and one that has hemophilia will be black in color, okay? So let's see. This one has hemophilia. So if I had to put it in, it'll be X with a capital H. Am I right? Capital H we said was, come on, capital H used for normal blood, and H being the hemophilia, so it's a small h. There we go. You've got to make sure that you concentrate and you give them everything. H, small h, and a Y, and this one will be an X, X. Simple, right? This one will be an X with an H and a Y. This one will be just a normal X and a Y. Remember, it's a capital H. These can be capital H's. Okay, this will be an X and an X, both capital H's. Okay, this will be an X with a capital H and a Y. This one will be an X and an X, both capital H's. This one will be an X with a small h and a Y. And this one will be an X, capital H, X, capital H. Okay, so I've just given you all the things. Oh, last one. This will be a male XY, and he'll be capital H. Okay, and then you just need to answer the question from the pedigree diagram. State the relationship between gender and hemophilia. Okay, most of the time, or all on this part, only the males are hemophiliacs. Okay, let's have a look. Write down all the possible genotypes for the individuals, Peter, let's see, where's Peter? We did it. Okay, so Peter, if I have a look at it, is X with a small h and a Y. Okay, where's the next one? There it is. It's two X's. It's a female. Okay, and they're both capitals. Okay, and the last one, parents, there it is. Oh, look how I made him. Oh, it is, it's right, sorry, I'm going mad. That was a female, hey? Yeah, and then this one, X, Y, and it's a capital letter. How cool is that? Okay, so that was quick and easy. I always call them the phylogenetic trees. They're actually quite cool, okay? Now, have a look at this one. This is normally a difficult one. Study the ones that follow, okay? Now, it gives you blue eyes for male and green eyes. Okay, blue eyes for male and female and all of that. Okay, so have a look at that. Write it all down. Copy it as quick as you can. Okay, once you have got that, I want you by yourself, because there's not going to be enough time. Hopefully I can go through it. Have you got it all down? If you copy that down, you must be able to tell me which has got blue eyes, which has got green eyes, and how you would know. Remember, in this case, if I have a look at it, how will I find out which is dominant and which is recessive? Let's see. Brown eye female. That's got a brown eye. This has got brown eye male. So it's got brown eyes. And out comes all brown eyes. Okay, so let's see. If they're all brown eyes come out, and I have a brown eye and I have that one, look what comes out. This is where you start looking at genetics. And we need to find out if this one is BB or if it's like that. Which is dominant? Is brown eyes dominant or is blue eyes dominant? Okay, so you've got to make sure that you concentrate and get them. So have you got all of that down? Okay, once you have got that down, I want you, by yourself, I want you to find out the genotypes of each and every individual there, A, B, C, D, and E. Don't get confused, because I've just made sure that you did. Have a look. Brown eyes for male and brown eyes for female. Okay? One is colored in, one is not. Make sure you concentrate when you do it. Don't get confused. And I will see you soon. Cheers.